So we are going to continue the session, and this session is on microglial phagocytosis and pathology. And our first speaker is Guy Brown from the University of Cambridge.
Thank you, Jenny. I'm going to be talking about microglial phagocytosis of neurons contributing to neurodegeneration. So you'll know that microglia are resident brain macrophages which specialize in phagocytosis, that is eating things, and microglia can eat excess synapses and neurons during development to shape neuronal networks. And microglia can protect the adult brain by eating pathogens such as bacteria, amyloids such as beta amyloid plaques, and dead cells and debris. However, excessive microglial phagocytosis of live neurons and synapses may contribute to neurodegeneration. It may do. That's what I'm going to be talking about. And you'll know that a lot of genome-wide association studies have linked Alzheimer's disease particularly to microglial phagocytosis. So a variety of genes that regulate microglial phagocytosis are genetically associated with Alzheimer's, but of course that doesn't tell us whether microglial phagocytosis is good or bad or both. So we need some evidence one way or the other. So microglia can phagocytose synapses, they can eat axonal arbor, they can eat axons themselves, they can eat the myelin wrapping of axons, and they can eat the dendritic arbor, but they can also eat the cell bodies of neurons, resulting in the death of the neurons. And we're interested in this because many neurodegenerative diseases, such as Alzheimer's, involve brain atrophy, which involves losing a large number of neurons. But brain atrophy doesn't occur is not accompanied by significant neuronal death, by which I mean there's not a large accumulation of apoptotic cells or necrotic cells, and there's little evidence that preventing neuronal death, for example, blocking apoptosis, prevents neurodegeneration. And therefore, there's a possibility that this loss of neurons could be due to microglial phagocytosis of live neurons, live but stressed neurons. And that's what I'm going to be talking about, and some of the evidence for that is outlined or reviewed here. So the first time we came across this phagocytosis of live cells was in cultures of primary microglia and astrocytes and neurons. And when we inflammatory activated the microglia with a variety of stimuli such as lipopolysaccharide or A-beta or tau or TNF-alpha, we saw microglial phagocytosis of neurons. So these phase-bright cells wandering around these cultures are the microglia, whereas these darker round things are the cell bodies of the neurons connected by neurites in the background. And occasionally you'll see a microglia phagocytosing a neuron. So for example, this microglia is going to phagocytose this neuronal cell body. And then it's going to eat this one, and then it goes up and eats this one, and then it comes over here and then it eats this one. But it's not surprising that microglia are phagocytosing neurons. What's surprising is that the neurons that are being eaten show no signs of apoptosis or necrosis. If we block apoptosis or necrosis by a variety of means, it doesn't prevent the neuronal loss, whereas if we block the phagocytosis, then we don't get the neuronal loss and the neurons stay alive. And we've investigated over a number of years the mechanisms. There's a variety of signals, including the stressing of the neurons causes flip of phosphatidylserine, which we know is reversible on neurons, which is driving the phagocytosis. So that's not good. I don't know whether that's because of the video here. Any ideas? It's gone completely grey. Wait for the program. Does anybody want to vote for the responses here? Oh, I've got 3. Restart the program, close the program, or wait for the program to respond. You can vote for your... Does the AV person have any? Restart. Okay, we'll go for that one.
Give you a few more minutes.
I don't know why it did that, but it's always used, it's often a disaster to put a video in a presentation. Okay, so we know a variety of the signaling that controls microglial phagocytosis of synapses and neurons. One of the major signals is the exposure of phosphatidylserine. But we know, and we know that neurons can reversibly expose phosphatidylserine, but that phagocytosis can be driven by many different signals and normally requires a variety of different signal changes. And the other major eat-me signal is desylation of neurons. So what that means is, so there are glycoproteins and glycolipids on the surface of neurons, and those are short sugar chains, and the terminal sugar is normally sialic acid. And that can be removed by sialidases, which exposes other sugar residues such as galactose or N-acetyl glucosamine or mannose, which can bind a variety of opsonins to drive phagocytosis. So terminal sialic acid inhibits phagocytosis normally by activating inhibitory C-GLP receptors, but when you remove that sialic acid, you expose other sugars that can drive phagocytosis. And David Allendorf, particularly in the lab, has shown that inflammatory activated microglia release a sialidase, that is an enzyme that can remove these terminal sialic acid residues, which is neuramidase 1, which is normally found in the lysosomes, and by lysosomal exocytosis, it's released from the microglia. This is just showing the release of this neuramidase 1, this very potent sialidase, which can remove these terminal sialic acid residues. And if you just add a sialidase to these co-cultures of glia and neurons, you can drive neuronal loss, which is just being quantified here. So this is just the neuronal density in these co-cultures of glia and neurons. And if you add sialidase, then you can lose neurons, and we know that's accompanied by microglial phagocytosis of the neurons. And if you block this phagocytic receptor, complement receptor 3, with an antibody, then you can block that loss of neurons. And you can also drive neuronal loss using, for example, LPS. You get a slow progressive loss of neurons in these cultures over 3 days or 7 days. And that can be prevented if you just block this sialidase with DANA, which is a sialic acid derivative, which blocks extracellular sialidases. So we believe that inflammatory activation of the microglia releases the sialidase, which desilates the surface of the microglia, as we've shown, but it also releases the sialidase into the medium, which can desilate the neurons, which drives microglial phagocytosis of neurons. Emily Kitchener in the lab has recently found that there may be a similar type of thing occurring with beta-galactosidase. So beta-galactosidase is normally found in a complex with neuramidase 1, super complex, which also contains a protective protein. And she's found that if you inflammatory activate the microglia, they release beta-galactosidase into the medium and onto the surface of the microglia, and that can potentially remove galactose residues, particularly from gangliosides, which are anti-inflammatory for microglia and are neuroprotective. LPS activation of microglia results in release of beta-galactosidase into the medium and active beta-galactosidase on the surface of the microglia. If you add extracellular beta-galactosidase, it causes neuronal loss, and LPS-induced neuronal loss is partially inhibited if you inhibit beta-galactosidase. So again, we believe that LPS activation and a variety of other forms of activation of microglia result in lysosomal exocytosis of a variety of things, including neuramidase 1 and beta-galactosidase, which are causing microglial activation and inducing microglial phagocytosis of neurons. Emily and Carl Reed in the lab have also shown that activation of microglia in a variety of different ways can cause release of chaperones from the endoplasmic reticulum into the extracellular space, including chireticulin and LRPAP1, also known as RAP, which causes some kind of alternative activation of microglia that blocks, so chireticulin and LRPAP1 can block phagocytosis by a variety of different means, and they also act as chaperones to block A-beta aggregation, and they therefore protect against A-beta-induced neuronal loss. But we've been interested in how you can prevent excessive microglial phagocytosis of synapses and neurons. And we've been through a variety of different phagocytic receptors to see if you knock them out in mice, whether they're protective or detrimental in a variety of brain pathologies. But it's microglial phagocytosis has both beneficial and detrimental roles, and therefore you have to look at the signaling, whether potentially that receptor might be involved in phagocytosis of live neurons. So in that context, we're interested in this P2Y6 receptor, which acts as an engulfment signal activated by the nucleotide uridine diphosphate generated from UTP, which is known to be released from activated or stressed neurons. And P2Y6 is expressed by a variety of different cells in the brain, but in the body, but within the brain, it's rather specifically expressed by microglia. We became interested in it when Inui and Kazumi published this paper in Nature showing that UDP is released by stressed neurons in vitro and in vivo and activates microglial phagocytosis via this P2Y6 receptor. And here they're just showing that if you add UDP to primary microglia, it increases phagocytosis about tenfold, and you can inhibit that with this inhibitor of the P2Y6 receptor, MRS-2578, or just by knockdown of P2Y6 in microglia. So we investigated whether if you added UDP to these co-cultures of microglia and neurons, you could drive microglial phagocytosis of live neurons. And this is just adding UDP to these co-cultures, and we're quantifying apoptotic, necrotic, or healthy neurons. And you lose healthy neurons actually relatively quickly after a few hours. And that can be prevented by inhibiting the P2Y6 receptor with MRS-2578, or by just removing the microglia from the cultures. So UDP is not directly toxic to neurons. It requires microglia, and it requires this engulfment receptor that is the receptor for UDP or microglia. This is in rat cultures. This similar thing occurs in mouse cultures. So if you add UDP, you lose neurons, but not if you take the cultures from P2Y6 knockout mice. And as I said, LPS causes a slow progressive loss of synapses and neurons in culture over a few days. And if you inhibit the P2Y6 receptor, then you can prevent this. You can also prevent it in cultures from P2Y6 knockout mice. What about in vivo? So we injected LPS into the striatum of rats, or one side of the striatum, and we could see increased microglial phagocytosis of neurons. But 2 weeks later, there was a loss of neurons on that side of the striatum, which was prevented by co-injecting an inhibitor of P2Y6. More recently, we used a more chronic model of inflammatory activation by injecting LPS into the peritoneum of mice on 4 successive days, and that's been shown to cause a relatively specific loss of dopaminergic neurons in the substantia nigra, and therefore is used as a model of Parkinson's disease. And that's what we're quantifying here, the tyrosine hydroxylase positive neurons in the substantia nigra pars compacta 2 weeks after injecting the last injection of LPS into the peritoneum of mice, and we lose neurons relatively specifically in the substantia nigra. But that's completely prevented in P2Y6 knockout mice. So if you don't have this microglial engulfment receptor, then you don't lose neurons. Now, we're using LPS partly just as a model of neuroinflammation, but it's possible that LPS is causally involved in neurodegeneration, because it's been shown, for example, by, not by our lab, but the lab of Caroline Williams-Gray in the Department of Clinical Neuroscience in Cambridge, that Parkinson patients have elevated levels of LPS endotoxin in their blood serum. So there's elevated levels of endotoxin in the blood of Parkinson patients, and that's been shown by 2 or 3 other independent groups. So there is more LPS in their blood, and that's probably because there's early gut dysfunction in Parkinson's patients. There's increased gut permeability and changes in their gut microbiome. But that's not specific to Parkinson's. It also occurs in Alzheimer's disease. So this is from this paper, but again, there are 2 or 3 other independent groups have shown an elevation of LPS in the blood of Alzheimer's patients. And that's interesting because LPS can synergize either with alpha-synuclein or A-beta or tau to cause neurodegeneration, and therefore there's a possibility that LPS could be causal in these diseases. A-beta also causes a slow progressive loss of neurons and synapses in culture, which can be prevented by inhibiting this P2Y6 receptor or knocking it out. And in vivo, we injected A-beta into the ventricles of mice and looked for uptake of neuronal nuclear material into the microglia. So the microglia are stained green here with an antibody against IBA1 conjugated to a green fluorophore. And we're also staining this is a hippocampal section, staining neuronal nuclei with an antibody against NUN. And when neuronal nuclear material is taken up into the microglia, then they stain yellow here. And 3 days after injecting A-beta into the ventricles of mice, there's a large increase in the uptake of neuronal nuclear material into the microglia in the CA3 region of the hippocampus, CA1 region, but also in cortical areas. There's a large increase in the amount of neuronal nuclear material taken up into microglia, but that's completely prevented in P2Y6 knockout mice. So if you don't have this engulfment receptor on the microglia, they're not phagocytosing neurons. And then we look for neuronal loss 2 weeks later in a variety of areas. So in prefrontal cortex, there's a loss of neurons, and in other cortical regions, and in the hippocampus CA3 and in CA1, there's a loss of neurons 2 weeks after injecting A-beta into the ventricles, which is prevented in P2Y6 knockout mice. So if you don't have this engulfment receptor, you don't lose neurons in response to A-beta. But that doesn't tell us whether this engulfment of neurons is good or bad, since damaged or dysfunctional neurons are potentially bad in terms of generating epilepsy or whatever. So we wanted to look at a functional readout. We used novel object recognition. That's putting 2 objects into the cage of the mouse, one of which is novel, and the mice tend to spend more time with the novel object if they recognize the non-novel object. But 2 weeks after injecting A-beta, they have a deficit in learning and memory, so they're more or less at 50% whether they interact with a novel or non-novel object. But there is a preservation in P2Y6 knockout mice, so A-beta does not induce any memory deficit in the P2Y6 knockout mice, so it appears to be beneficial blocking this engulfment receptor in this particular model. So we also wanted to look at a tau model, a chronic tau model. So we used P3ONS tau expressing mice. So P3ONS tau is a variant that can drive frontal temporal dementia, but here we're just using it as a model of tauopathy in mice. And this was together with Maria Spillantini, who developed these particular mice. And we crossed these with the P2Y6 knockout mice on the same genetic background. And then we looked for neuronal loss at 7 months in this particular genetic background. But there's very subtle neuronal loss in the perirhinal cortex here and in motor cortex, but it's a relatively small amount of neuronal loss, but it's reduced by crossing these P3ONS tau mice to P2Y6 knockout mice. So there's reduced neuronal loss. And there is protection against the memory deficit that develops in these mice. So at 7 months, the P3ONS tau expressing mice have a deficit in learning and memory, which is prevented by crossing them to P2Y6 knockout mice. I should say that the pathology in the spinal cord in this mice is not affected at all in terms of time course by P2Y6 knockout. So that we don't know what that means. It may mean that the pathology is different in the spinal cord or just because it's more severe. But there is strong protection in the brain. So in summary, I've told you that one mechanism of neuronal loss during neurodegeneration may be microglial phagocytosis, and this excessive microglial phagocytosis of neurons may be blocked by inhibition of the P2Y6 receptor. So together with the drug discovery institutes of UCL and Cambridge, we're trying to develop novel P2Y6 antagonists to see whether that can be protective. And this work was largely done by Mar Puig-Delabole and Stéphane Mildet with the help of Anna Villalta and David Allendorf and Claire Butler, together with Jake Dundee, Emily Kitchener and Kyle Reed, and a variety of other people here, including particularly Katerina Papushenko from Vilma's lab. Thank you.
Thank you, Guy. Questions, anybody? Anybody got a question? Yeah, at the back.
Yeah, really interesting. I might have missed this, but I'm a little bit wondering about the link between the UDP extracellular release that activates the microglia so they can start phagocytosing neurons, and then how that would specifically get rid of the stressed neurons in the in vivo situation. Because the way that you show your cultures, those microglia are then able to clear away any cell, any neuron. So is it like extremely short range, or is there a chemical gradient, or does it adhere to the surface of the cell? What gives them that specificity so that they're only getting rid of the cells that are producing it rather than getting activated and just clearing every neuron around them?
Okay, yeah, good question. So Inui and Kazumi showed that stress neurons release UTP, which is converted to UDP by by nucleases in the medium. And those nucleases are very active, and potentially would then degrade the UDP further, so that the UDP wouldn't be able to diffuse very far from the site of its release. So they showed that stress neurons release UDP locally to act as a local stimulus for phagocytosis. So they use a variety of stimuli, but we've also used a beta can cause UDP release from live neurons. So the idea is that UDP would be released locally from stressed neurons and would be a local stimulus to the surrounding microglia to cause phagocytosis. And the idea would be that various types of nuclease in the extracellular medium would limit the diffusion of the UDP.
Any question here?
Yeah, well, very nice talk. Actually, there are lots of the UTP and receptors in the retina, including the P2Y6 you used. So my question is whether the different UTP receptors have different functions in the brain.
So your question is whether UDP receptors have different function in the retina?
In the brain?
In the brain, I mean, in the microglia.
Yeah. I don't know about the role of P2Y6 in the retina. But again, it would require stress neurons in the retina acting on the microglia. And I don't know whether that would result in phagocytosis of the neurons or bits of neurons.
Okay, thank you.
But Jake has shown that this can also have a role in microglial phagocytosis of synapses as well as neurons. But he'll be talking about that later.
Okay, any more questions? I've just got one. I mean, I think it's back to the first question. If you've got UDP being released from stress cells and then you block P2Y6 and you say you're not getting the neuronal loss, so does that stressed cell recover?
Well, that's a good question. In culture, those neurons, if you block the P2Y6 receptor or if you knock it out, those neurons survive and appear functional as long as you can culture them. And in vivo, the neurons stay alive, but we don't know the extent to which they're functional or dysfunctional. But when we look at particular pathological models and we cross it to P2Y6 knockout, it's beneficial in the ways that we've looked at, such as novel object recognition.
Okay. All right. Thanks very much, Guy.